Researchers discover a new link between muscle signals and bone loss. Exercise causes muscles to produce a chemical signal that helps prevent bone damage, but as bones age, they start losing their ability to understand that signal, a research team led by Indiana University School of Medicine scientists has found. The discovery may help explain why people are more susceptible to bone loss as they age. It may also lead to interventions that work around or even prevent older bones' inability to recognize the signals the muscles are sending, one of which might just be more exercise. The research team, led by Linda Bonewald, PhD, director of the Indiana Center for Musculoskeletal Health, discovered that a metabolite produced by muscles during exercise, known as LBA-IBA, helps protect osteocytes, the bone cells that oversee bone health, repair, and renewal. But the researchers reported in a recent edition of the scientific journal Cell Reports, as the osteocytes age, they lose their ability to respond to the LBA-IBA. The culprit, the researchers found, is the receptor on the membranes of osteocytes that interacts with the LBA-IBA molecules produced by the muscles. Experiments using mice as well as muscle tissues in the lab demonstrated that both younger and older muscle tissues produced LBA-IBA. However, examination of younger and older bone tissues revealed that the receptor needed to interact with LBA-IBA was largely missing in the older osteocyte cells. If you're a young person and you exercise, you get the benefits. You get good increase in muscle, you get good increase in bone, the whole bit, said Dr. Bonewald, professor of anatomy and cell biology and orthopedic surgery. As we age, you have less of an effect of exercise. We're losing expression of the receptor in our bone, and that's likely one reason why our bones don't respond well to exercise. Having identified the role of LBA-IBA, the researchers then asked, how does the presence of the metabolite protect the osteocytes in the bone? The answer is in the mitochondria, tiny organelles, many organs, inside cells that play key roles in cell function, such as energy production and cell division. Mitochondria are susceptible to breakdown by free radicals or receptive oxygen species that are more prevalent in the body as people age. LBA-IBA prevents the breakdown caused by the free radicals and thus protects the osteocytes, the researchers found. The discovery suggests potential routes to therapies that might counteract the changes that reduce the protective effects of LBA-IBA, Dr. Bonewald said. That could include alternative methods for blocking the destructive effects of free radicals on the mitochondria. We think if we can keep the mitochondria healthy, we'll keep the osteocytes healthy, she said. In addition, experiments now are underway to see whether continued exercise in older mice can help maintain production of the receptor that was missing in the older osteocyte cells. As they age, mice become more and more sedentary, just like humans do. So if we can keep them active, maybe we can level out the receptor expression, Dr. Bonewald said. Discussions have begun with researchers who conduct clinical trials, such as exercise studies in people with osteoporosis, to develop ways to measure levels of LBA-IBA in humans. That could lead to a variety of clinical trials testing new ways to protect the integrity of bones as people age. In addition to Dr. Bonewald, scientists contributing to the research were Yukiko Kitase of the IU School of Medicine, Julian Vallejo, William Guthal, and Harika Vemula of the University of Missouri, Kansas City, Katrina John of the University Medical Center for Hamburg-Eppendorf, Hamburg, Germany, among others.